Good morning. Biology 1302, Chapter 11. You may notice this is a different room. I'm on the main campus today, but this is Chapter 11 of Biology 1302. And this is going to be DNA technology, the last of the third unit, the unit on DNA inheritance and biotechnology. DNA technology has entered basically every phase of our lives. This is a gentleman right here. I'm sure if you look in your text, you will see that this is a man who probably served years in prison for a crime he did not commit, and ultimately he was exonerated by DNA technology. So it's basically invaded is not a good word. It's become involved in every phase of our life. We use it in research, medicine, agriculture, criminal justice, and many other fields. DNA is found everywhere, and DNA technology is found everywhere. Scientists now have the capability of manipulating DNA in many different ways. In certain instances, we can switch genes on, we can turn genes off. We can take genes from one organism, put them into another organism. We can take genes from one species and put them into a different species. So all kinds of stuff we can do. And anytime you're manipulating DNA, we refer to that as DNA technology. We can change DNA. The first thing we have to be able to do is to read these sequences. You have to be able to read the sequence. That is used to identify individuals. You can manipulate it. You can put it in test tubes. You can put it in living cells. You can change it in entire organisms. We now have the capability of doing lots of things that we couldn't do just, say, 10 or 15 years ago. Again, mastering the concepts of DNA technology, what field it's useful in. Basically, if you can name it, we can use it. If it's research, if it's developing new medications, if it's developing new treatments, even up and to include the possibility of bringing extinct species back to life. There is work right now being done to try to bring the woolly mammoth back from extinction using DNA technology. We use a variety of tools to do this. We can use small pieces of DNA in test tubes. We can use entire genes or the entire genetic makeup of a cell. We now have the technology to manipulate all of those things. You can see these in the Petco and the PetSmart now. They're glow-in-the-dark fish. These are fish that do not appear in nature. They have been developed through DNA technology. And what scientists have done is they have taken the bioluminescent genes out of certain jellyfish and some other creatures and inserted them into the fish genome. So we have fish now that glow in the dark. Bacteria can be engineered to manipulate DNA. Bacteria can produce desired molecules, especially drugs. Insulin, the treatment for type 1 diabetes. Historically, insulin came from pigs. If you were a diabetic 40 years ago and you were injecting yourself 3 or 4 times a day with insulin, that insulin came from pigs. And there were a couple of problems with getting insulin from swine, and that is, number one, it's a very labor-intensive process. Actually, there's about 3 problems. It's labor-intensive to get. The possibility of contamination is high. And then we have a segment of our community who may not want to use that. Think of folks of the Jewish faith or folks that are followers of Islam. They may not feel comfortable using a pork or a swine-produced product. I've had students in the past in A&P lab that once we started doing the fetal pigs have wanted to opt out of the dissections because of fetal pig dissections. They did not want to do that because it was against their particular beliefs. and I respect that. I gave them other things that they could do. But insulin is actually produced using a bacteria. I'm not sure how well you can tell, but if you actually open up these PowerPoints, you can see it. This chromosomal material, this DNA right here, is actually two different colors. There's a dark blue segment and there is a light blue segment. Well, the dark blue segment is the original DNA found within that bacteria. The light blue is a gene from something else. And just in my example of insulin, that gene is the gene that produces insulin in human beings. We have the technology, we've been doing this for several years. We can take that human gene, we can splice it into the genome of this bacteria. Every time this bacteria reproduces, it produces more copies of that gene. And then every time that cell is doing the things it's supposed to be doing, when this DNA is producing the proteins that this cell needs for its everyday survival, it's also producing insulin. This bacteria doesn't need insulin, so it packages that insulin and ships it outside of the cell. It basically treats the insulin as a waste product. The waste product then collects in the environment, usually in a bottled broth media, and then the insulin is captured and clarified and purified and put in little vials and put into the pharmacy for our friends with type 1 diabetes. Another one I want to point out here, there are other bacteria who have been engineered to degrade petroleum, pesticides, other soil pollutants. There are also bacteria that have been engineered to consume explosives and render those explosives void or inert, so they don't cause explosions anymore. The structure within that within the bacteria that does this is called a plasmid, and here's my dark blue DNA material. A plasmid is a small circle of double-stranded DNA that separates from the chromosomes within bacteria. So the bacteria will have its normal chromosomal material, and then it'll have this extra little piece of bacteria called a plasmid. What we can do is we can take those plasmids, open them up at specific sites, and insert our gene. In my example, I used a gene for producing human insulin. Once we have that accomplished, here's my DNA from the human cell for making insulin. We find the plasmid, we open the plasmid at a particular place, we segment the DNA, we find the portion of that uses or that makes insulin, and then we stick it into the plasmid. We do this by using structures called restriction enzymes. These restriction enzymes are going to cut the DNA at a very particular place. I don't know how well you can see that, but this says CTTAA and CTTAA. So this restriction enzyme is engineered to cut at that particular place. If it cuts this one at that place, it's going to cut this one at the same place. And then these little tags, or these little long ends that are left after the enzyme is cut, those are called sticky ends. Several ways to move plasma into host cells. We can zap them with electricity. We can use something called a gene gun that shoots DNA directly into cells. These are used almost exclusively in plants. The gene gun is an almost exclusive plant technique. We can use membrane bubbles that fuse with the recipient cell, or we can use a virus to infect the recipient cell. So several different ways to get the new DNA to insert itself into the original plasmid DNA. Recombinant plasmids combine plant chromosomes when plant cells are infected by genetically modified bacteria. So here's the herbicide-resistant gene right there in the bacteria. There's the bacterial chromosome. This is some agricultural designed bacteria. Here's the normal chromosome, the unaltered cell, and it just happens to be a tobacco cell that they've drawn. And you can see here's the chromosome. We infect this line of cells or this plant with this virus or this bacteria, excuse me. And what we ultimately get, you will see on this gene right here, you now have this little piece of sort of orange colored chromosome. That gene has been placed right there. So now this plant is resistant to a particular herbicide. So as the farmer puts the cotton into the ground and the cotton starts to grow up and weeds start to grow in around the cotton, traditionally, after a certain point, all the farmer could do was go out there with a hoe and chop out the weeds. There was no way to spray it, because if he sprayed the herbicide onto the plants, it would not only kill the weeds, but it would kill the tobacco plant as well. But this new strain of tobacco, the farmer can now spray with the herbicide. It will kill the weeds, but it will not harm his tobacco plants. Again, just a little more detail. You see that the transgenic cell divides, producing more and more cells, and eventually you have a tobacco plant here that is transgenic, and it is now resistant to the particular herbicide they are talking about. The fish, again, we showed earlier, expressing those jellyfish genes for glowing, and it's done in basically the same way. Scientists also use viruses to create transgenic animals. We've been hearing for the last year, just so happens the day I'm recording this is the 1-year anniversary of the first case of COVID found in Arkansas. We are hearing lots about viruses today, but all the news about viruses is not bad. There is some good news about viruses. Genetically modified viruses can carry desired genes. It can be used to infect early embryonic cells, and it can take an animal that was going to be very susceptible to something, less susceptible or possibly even resistant to that thing. When cells divide by mitosis, as animal grows and develops, they pass the recombinant DNA to all the cells in the animal. DNA sequencing reveals the order of the bases. When we talk about the DNA sequence, what we are talking about is the series of bases, the A's, the C's, the T's, and the G's that make up that particular strain of DNA. And you'll notice here, you see a peak chart showing the different bases. This is what you're more commonly going to see, is the A's, the C's, the T's, and G's. It's useful for deducing protein sequences and for determining evolutionary relationships among species. Let's say this is a stretch or a sequence of a particular gene that multiple different species share. Multiple species share this gene. And what we would do to determine the relatedness of those species, let's say there's 7 or 8 different species of tiger, and we look at all those tigers, and what we would look at is this sequence and how closely is it in relationship to the other tigers. Let's say we have the Bengal tiger and the Siberian tiger, and they both have this same sequence, but the Bengal tiger, while it has a T right here, the Siberian tiger has an A right there. That's the only difference in this link. That signifies that those two tigers are very closely related. But let's say the Bengal tiger and the Javan tiger, instead of having this be a T, this is an A, and this is a T, and this is a T. So now you have, instead of one difference, you have 3 differences. The more differences, the more distantly related those organisms would be. Our technology has improved DNA sequencing significantly. Historically, we were pretty limited in what we could do with DNA. If you watch the crime drama TV shows, I'm a fan of the crime drama TV shows, And one of the things they used to do back in the late 80s and early 90s when DNA technology was first starting, every so often there would be at one of these shows, and they would have a DNA sample that was found at a crime scene. But they couldn't analyze the DNA because it would destroy the entire sample. And I don't know how much you know about the way court, criminal cases go, but if the prosecution has some physical evidence, the defense has to have access to that, that evidence as well. If it's a bloody shirt, the defense has to be able to at least look at that bloody shirt. If it's a blood sample or a DNA sample and the prosecution is going to test it, then the defense has to have the opportunity to find their own lab and have it tested on their own. And so if one test would completely destroy the sample, you couldn't use the sample, you couldn't test it. Because then once you tested it, it might prove that defendant A was the killer, but you couldn't use that evidence in court because the defense was not able to use it. Well, we no longer have that problem because now we have technology that's more reliable, faster, and cheaper to produce lots of DNA all at once. The cost of sequencing the human genome plunged from about $100 million in 2001 to about $1,000 in 2017. And it's even less than that now, because how many of you, either you yourself or know somebody who's paid $99 down at the Target and sent away to 23andMe or that other place to find out what your genetic code is? In 2001, that process would have cost you $100 million. In 2017, it would have cost you around $1,000. You can now do it for less than $100. They will sequence your entire genome. Not only that, for an extra $100, they'll tell you what if you have any genes that may lead to health issues, like you may have a mutation in the BRCA1 gene, which could lead to breast cancer, and there are other things that can be shown up as well. PCR, polymerase chain reaction, is the original technique for use for rapidly producing large amounts of DNA. It is done in a test tube. It gives investigators, again, enough DNA to analyze. Basically, what you do is you take the DNA, you take DNA polymerases, you take nucleotides, you take primers, and a target DNA sequence. Here's your target sequence right here. You throw it in a tube. You put these primers, you put the nucleotides, and you put DNA polymerase in there, and the DNA polymerase, along with the primers, will attach themselves to here's my sample DNA. You see it says raise the temperature, the DNA separates, and then the primers hook on the DNA polymerase, and we start making more, and that will just continue to make more and more and more. I did some of this when I was over at UALR taking some graduate school courses. The machine that they had over there and again, we're talking the late 90s, so this was kind of early technology. The machine for doing PCR it looked kind of like a toaster oven. If you have a toaster oven in your house, that's the one that you can make toast with, or you can set it at a different temperature and, bake, make some hot pockets or pizza pockets or those kind of things. It can be used as a little small oven. It kind of looked like that. It was much more involved. The big thing with PCR is temperature. You raise the temperature to separate this, then you lower the temperature, and the new DNA starts to form. Then you raise the temperature again, and this new strand will separate. So what you have is a cycle of raising the temperature and lowering the temperature, and raising the temperature and lowering the temperature. And so that's what set that PCR oven apart from a typical, oven that you would have, a toaster oven. But it looked a whole lot like a toaster oven. Again, these copies are just separated. Again, as you raise the temperature, each strand will separate. The primers and the DNA polymerases will attach. And again, you start with 1 copy, you get 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and you can create millions of copies of this in just a matter of a few minutes. It goes very, very quickly. As I recall, I think, and again, we're talking 25 years ago, so my memory may be fuzzy, but I think we set our machine with our sample DNA to make for 20 minutes. And after 20 minutes, we had enough DNA that you could literally shake the tube and see the clusters of DNA. It looked kind of like a white sort of foaminess in the broth that was growing, and that was the DNA. PCR is used in DNA profiling. Once they have enough DNA, they can detect genetic differences between individuals. PCR copies highly variable target sequences throughout the genome. Here, for instance, is chromosome 5. It's going to take certain segments of chromosome 5 and start to replicate them. They will then be measured on a bar chart, and you will look at short tandem repeats. If you've ever seen the Maury Povich show when he does the Who's the Daddy, that's what they're doing. They're doing a DNA analysis of short tandem repeats, and if more short tandem repeats appear in the baby and one of the prospective fathers, that's where they get that alignment. DNA profiling again shows how many short tandem repeats there are in a particular person's genome. This person has the short tandem repeats here, so you get this graph. This person has them here, so you get this graph, and then this graph. And again, here's the DNA from the crime scene. So we see a spot at 10 and 11, at 17 and 19, at 20 and 24. This particular gene at 6 and 8. But as you can see, man number 2, you're a perfect match for the crime scene DNA. You, sir, are going to jail. They compare the SPR profile, again, short tandem repeats, of an individual with a profile from the reference, reveals that the 2 samples are a match. Again, the number of possible repeats is 6 to 18, 6 and 8, 7 and 9, 6 and 10, 6 and 8. Dude number 2, enjoy prison. Again, this is how this gentleman was exonerated and released from prison. Due to DNA profiling, the number of innocent and wrongly convicted people being exonerated and released from prison has been greatly increased. You can see this started in the early 90s. It gained traction, and starting in the early 2000s, you see there's DNA exonerations in the United States. It's been hovering right around 60 every year since then, every series of years. And basically, this is a number that's going to continue to be like it is. Again, as DNA technology improves and our ability to speedily run these samples, this can get, the reason a lot of this was, these numbers were really low back in this time is the labs that were capable of doing this type of analysis were really backlogged, and they were looking at current crimes, and the exoneration of people wrongly convicted, right or wrong, did not get high priority. But now there are so many labs that are able to do this technology, and they can do it so quickly that the numbers game is starting to catch up, and we can do this. And hopefully in the future, we will have far fewer innocent people going to prison because they're eliminated on the front end. Before trial even comes up, we have the evidence that, well, somebody may have been murdered at a robbery, and a particular person may have been seen coming out of that store at the approximate time of the robbery. Historically, that person probably would have wound up in jail or in prison because of the eyewitness testimony placing them at the scene at the time. But now with DNA and the store owner has been stabbed with a knife, and the attacker cut himself with the knife, and his blood is on the dead body, and we take his blood and we go, well, even though this guy walked into the convenience store at approximately the same time the crime occurred, he just walked in, looked around, didn't see the owner, and turned around and walked back out. The owner was laying behind the counter having been stabbed 47 times, but that person didn't do it. Historically, that person probably would have wound up in prison, especially if a passerby had seen him coming out of the store, or he had passed somebody going in the store and the second person in the store had gone in and looked and found the body. But now they can be exonerated on the front end and they never have to spend any time in prison. And hopefully, as the years go by, we will get better and better at using DNA technology on the front end to eliminate suspects, not just to include them. Stem cells and cloning, add new ways to copy cells. Lots of controversy about stem cell research. Stem cell research often sparks public debate. Some see it as useful technology, others see it as unethical. I think it really depends on what your idea of unethical is. And again, we could talk at great length, which we're not going to, about the different things we can do with stem cells. But the discourse over stem cells and cloning combines science, philosophy, religion, and politics. Stem cells are undifferentiated. A stem cell, what a stem cell is, any cell in your body that has the capability of making a copy of itself and any other cell is classified as a stem cell. The cells that make your blood cells, the cells responsible for making red blood cells, white blood cells, and platelets, they are stem cells. If you ever hear someone refer to adult stem cells, that's the cells they're talking about. When someone is given a bone marrow transplant, what they're actually getting is a stem cell transplant. They bore into my hip bone, take my bone marrow, which contains lots of these cells, these primordial blood cells, and put them into someone else, and then that someone else possibly can start to develop good blood cells. Maybe they have leukemia, where they were making bad blood cells in the past. Excuse me. Each one of those primordial cells has the ability to either make a red blood cell, a white blood cell, a platelet, or more of the original cells. Therefore, it's a stem cell. In this drawing, whoops, the stem cell is this early embryonic cell. Here's my fertilized egg, comes down here. These are the stem cells. These cells are making these early embryonic cells. And during the first 5 to 6 days after fertilization, these cells are simply making more copies of themselves. You know, 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 32, 64, 128, 256, 512, 1024. It just keeps replicating that way. But after about 6 days or so, these cells start to differentiate. And some of these cells will then become nerve cells. Some of them will become muscle cells. Some of them, in this drawing, become red blood cells. So as these stem cells, and for a while, these stem cells, in addition to some of them making these, some of them are still making more of these. Eventually, we get to a point where all we are making is these specialized cells. Again, stem cells, embryonic stem cells, come from very early embryos and give rise to cell types in the body. Again, one of the issues with stem cell research is, where does it come from? Where do they come from? One of the pieces of misinformation that has been cast out for years was that these stem cells came from abortion clinics. And that's not where they come from. They generally come from what are called fertility clinics. Now, a fertility clinic is not an abortion clinic. A fertility clinic is a place where couples go that can't have a baby the old-fashioned way. They go to have what is used to be, and I'm not sure this is a proper term anymore, so nobody be offended when I use this term. This is just a historic use of the term. They were called test tube babies. Dad's sperm and mom's eggs would be fertilized outside the womb. And then the fertilized eggs would be then placed into the mother's womb in the hope that they would attach and grow. I have a niece. My oldest brother's daughter was born via in vitro fertilization. She is a test tube baby. I would never call her that. She's my niece, but that was the way she was conceived. And what happens in these cases is when my brother and his wife went in to have their procedure, they didn't just fertilize one egg. They fertilized a bunch of eggs, and we're going to talk about maybe dozens of eggs. And they got dozens of eggs to this stage of development, to just before they started differentiating. It was still just a big ball of cells. And at that point, those embryos get frozen. And again, the technology to do this is way up over my head. But those are frozen and they're kept in storage because this is not a perfect procedure. Just because they implant 5 or 6 eggs into a woman's womb doesn't mean they're going to implant and grow. So the couple may have to go through this process 3 or 4 times. But once they get the baby they want, they still got maybe 100 of these in a storage facility. Well, that company is not going to store it for free. So what do? Well, they have to decide, do I want to keep my eggs or do I want to get rid of my eggs? And so the place, the lab will ask the couple, well, since you no longer want your eggs, do you want us to A, destroy them, or B, to sell them to science? And that's the couple's decision. If they sell them to science, that's where stem cell research comes from. There's not, I saw all the editorial cartoons in the early 2000s where there was some shady looking character in a hat and an overcoat and hiding himself with a bucket at the back door of the abortion clinic. Wait, yeah, that's not what happens. That is not what happens. Adult stem cells, as I mentioned before, are found in the bone marrow. They come from adult tissues and give rise to many, but not all, types of cells in the body. But again, they can be used very similarly to embryonic stem cells. Some researchers say for certain fields of study that the adult stem cells are actually better than embryonic stem cells. Cloning creates identical copies of an organism. Cloning, also known as asexual reproduction, is common in nature. These are different plantlets. There are particular plants that will produce by cloning. You see it in bacteria, archaea, many protists. Plants are very easy to clone in the laboratory. Plants will clone themselves in nature. Anytime you see a plant that grows from the root system of an older plant, that is an example of a type of cloning. Somatic cell nuclear transfer is used to clone animals. Remember from previous, somatic cells, those are those cells that are not sex cells for human beings. Chromosomes 1 through chromosome 22 are all somatic cells. This is an incorrect statement. The first cloned animal was Dolly the sheep. That is an incorrect statement. Dolly the sheep was not the first cloned animal. Dolly the sheep was the first cloned mammal. We have been, for about 20 years before Dolly, we have been able to clone frogs and salamanders and some other amphibians. But Dolly was the first sheep. Ultimately, what they did to create Dolly was they took the nucleus from an adult sheep cell and was used to create Dolly, whose DNA was identical to her donor. The diploid nucleus from an adult donor cell is used to replace the haploid nucleus in an egg. So we take an egg. This egg contains only half the genetic material it's supposed to have. because it is an egg. If for a human, it would be 23 chromosomes instead of 46. We take the nucleus from another organism, our donor, in this case, Dolly's, not mother, but clone. They took the nucleus from that egg, placed it into the egg. Nope, they took the nucleus from, they actually used udder cells, cells from the sheep's udder. That's the area down under where the milk comes from. The nucleus of the udder cell was placed into the egg where the egg nucleus had been removed. They then implanted the surrogate mother animal. The cell then goes through mitosis and development, and when the new animal was born, it was a clone of the adult donor animal. They don't have pictures here, but one of the things they did to ensure that this process had worked quickly is there's two different types of sheep. There are two different species of sheep, but they're very closely related and they can breed across term. One of them has a black face, the other one has a white face, and they are, you know, very, very ingeniously called the white-faced sheep and the black-faced sheep. Dolly was a white-faced sheep. They took this udders from a white-faced sheep, but they implanted it into the black-faced sheep mother. Then she brought the baby to term and gave birth to Dolly, and Dolly was born with a white face. So they were relatively certain at that point that their technique had worked properly. They then went in and did some DNA analysis to make sure it had worked, and in fact it had. Is human cloning ethical? It could help infertile parents have children. Could also be used to harvest embryonic stem cells. There are lots of ethical issues. You may have read on the internet where supposedly there have been human cloning experiments being done in certain parts of the world. I won't mention any countries, but you know, it is supposedly happening. I don't know whether it is or not. I'm not real sure what I feel about cloning. Again, this one up here, human cloning could help infertile, and I probably should say infertile parent, because I mean, I guess if I'm an infertile male and I'm married to a fertile female, but if I'm making a clone, that clone is not a combination of her and I. It's a combination. It's me or her. So again, that's a little confusing, that statement there. But it could help people have children that might otherwise not be able to. Maybe you have a woman who is for whatever reason doesn't produce eggs, or had some medical condition and had to undergo a hysterectomy very early in life for some medical reason, and now possibly, you know, but again, you have to use a surrogate mother for that, because if she had to have all of her female parts removed, she wouldn't have the parts to make a baby, to gestate a baby anyway. But again, lots of ethical questions. My sister-in-law, who has passed away a few years ago, was always concerned about the soul. Would a human clone have a soul? Since it was not the combination of a man and a woman, it didn't occur naturally. And since if I cloned myself, for instance, my clone would be an exact DNA replica of me, even though it's 50 years apart. Would that person have a soul, was her concern. Again, that's philosophical, and it's, again, way above my head. I'm just at the point where I don't think we ought to be cloning humans at this point. Oh, the other thing about human cloning is, and it may have mentioned it here, no, I don't see it, but it could also possibly somebody who needed a kidney transplant, and you know, well, if you don't get a kidney, you're going to die in 2 years. Well, I'll clone myself, and that clone will have 2 good kidneys. As a matter of fact, there was a science fiction movie made back in the 80s or 90s. I cannot remember the name of it, but it was basically, that's what they were doing. They were, rich people were growing clones for extra body parts for themselves. They had developed a technique where these bodies were being reared in a vat of material. They had engineered so the body never developed a head, so it was just a body, and it was just all the organs you would need. And of course, I guess you didn't get eyes or ears, but you could get hearts and lungs and kidneys from your clone. Again, I wish I could. Every semester I say I'm going to look back and see if I can figure out the name of that movie, and I never think to do it. DNA probes are used to detect particular sequences. We have a fluorescent label right here that recognizes a particular strain or a particular sequence of DNA. We put it right here, we mark it. DNA probe can be used to test whether a patient inherited a cystic fibrosis allele. We have a probe, we look for that sequence, and the probe binds at that sequence, and then see it lights up right here showing that whoever this person is, they have the cystic fibrosis gene. They can test for mutations in an embryo, a child, or an adult. Again, these are things that we can test for using DNA probes while the child is in the very early stages of development. We can tell in the first trimester if someone will be born with cystic fibrosis, hemophilia, Tay-Sachs disease, sickle cell, and there are many, many others as well that we can determine. Pre-implantation genetic diagnosis is used to help reduce the odds of having a child with a genetic disease. First, a man's sperm fertilizes several of a woman's eggs in a laboratory dish. This is called in vitro fertilization. These are the fertilized eggs. Then the researchers extract one of those eggs. They extract the DNA. They use DNA probes to search for harmful alleles. And DNA from children to adults can be screened in the same way to identify existing genetic diseases before symptoms occur. What we're talking about here is before these eggs would be implanted, they would test one, and if they found that this baby was going to be born with cystic fibrosis, hemophilia, and sickle cell, they would probably counsel this couple not to have children. But you could take skin cells, for instance, from children or adults, do the same type of screening, and find out, do I have that BRCA1 mutation that causes breast cancer? Gene therapy can be used to treat diseases. It may someday provide new treatment options for genetic diseases. The overall goal is to supplement a faulty gene with a normal, healthy gene. This person has cystic fibrosis, which is caused by a mutation on chromosome number 7. The mutation occurs, the cystic fibrosis protein is produced, but through gene therapy, we could produce a virus that has the good gene. We could inhale the virus, carries those good genes into cells. Those cells start to grow, produce the normal gene, and possibly over time, most of these defective lung cells would be replaced by good lung cells, and the cystic fibrosis could be cured or its effects greatly reduced. And again, cystic fibrosis is caused by a mutation on gene 7 or chromosome 7. We talked about that. There's the whole process. Again, lung cells now produce the normal protein. CRISPR is a way of cutting genes and edit specific genes. You will see lots of the things about it as you read, especially those of you who are heading into more advanced biology classes. If you take genetics class, you will learn about CRISPR. But basically, it determines a segment of DNA, opens that segment, and produces the new protein. Then it turns it off, or it can induct something in there, so the system will stop. Technique allows researchers to quickly and easily modify one specific gene in an organism without affecting the other parts of the genome. Some uses of DNA technology, again, may raise ethical issues. DNA technology in medicine can reduce human suffering by improving the chance of having healthy children, detecting disease early, offering the prospect of new treatments. But how should it be regulated? Here's the big thing that virtually everybody agrees with. If I had a test, say in my 20s, that showed that I was going to develop certain health issues later in life, could that information be used against me in maybe hiring practices for a job? The company looks at me and says, well, yeah, well, you're more than likely going to develop cancer within the next 10 years. We don't want to hire you. Or the insurance company says, well, you look like you're going to develop cancer in the next 10 years. We're not going to write you a policy. So how is it regulated? Weeds, getting a boost from transgenic plants. Genes from transgenic crops can be transferred to weeds by natural pollination. Offspring are so-called superweeds. They germinate faster, produce more seeds than weeds without the gene. So here you see normal weeds and the weed with the booster gene. And then here's what happens. This is the number of seeds germinating, and this is the number of seeds for plants. So you see the weeds with the booster gene. Not only do they germinate at a higher rate, but they produce more seeds. Again, this is the concern a lot of people have with what are called genetically modified organisms, is can those modifications cross over into native populations? If that happens, for instance, the Monsanto Corporation now has a particular type of soybean, and the soybean is resistant to Roundup. Roundup's the #1 herbicide on the planet, I would guess. But you can spray Roundup on this particular type of soybean and it doesn't kill it. So if you've got all of these weeds growing around your soybeans, you can just fly a crop duster over, drop Roundup on it, and it kills everything but the soybeans. But what happens if that gene from the soybean is able to cross into the weeds through cross-pollination? If that happens, then the weeds will start to produce, and now not only are they having a larger than normal germination rate, they're also producing a whole lot more seeds, and they're going to be resistant to Roundup. So now we're going to have to develop a whole different product to kill the weeds without killing the soybeans. So all types of issues with genetically modified organisms. All right. Questions, comments, or complaints, please send them to me, trussell@uaptc.edu, or as always, you can use the use the contact tab or contact link, course messenger tab within Blackboard. Thank you. It's Thursday where I am, so everybody have a good weekend.